Hello, I'm Chris Steffen,
I'm Indy Rice.
and for our SHIRT program, we work up here at the NCRC, North Campus Research Complex.
Here, scientists across many disciplines collaborate on groundbreaking research.
For our SHIRT program, we worked under Dr. Tim Bruns and as part of the P Neuro Lab.
Which is the Peripheral Neural Engineering and Neurodynamics Lab. What does peripheral neural engineering mean? Often, when people think of the nervous system, they focus on the brain. However, our research targets the nerves that originate from the spinal cord and go into the periphery. Peripheral nerve stimulation can be used to treat a variety of disorders, including pelvic dysfunction. While the P Neuro Lab usually focuses on bladder function, I've been working on a study to address female sexual dysfunction. Over 25% of women suffer from sexual dysfunction, compared to only 15% of men. And yet there's a multi-billion-dollar erectile dysfunction drug industry and still no reliable option for women. We hope to develop the use of peripheral nerve stimulation to help address female sexual dysfunction. How does this work? Nerves that originate from the spinal cord can have both motor and sensory branches. Neurons within these nerves communicate with changes in their ions, which we can hijack with electrical stimulation. With the right stimulation parameters, we can activate the sensory nerves of sexual organs. This may drive a spinal cord circuit for an arousal response. The arousal response that is easiest to measure is blood flow. This is a graph of baseline blood flow, but with the stimulation period shown with the gray box, blood flow can increase. During steady arousal, blood flow continues to fluctuate, even after stimulation. So, our research indicates that stimulating these nerves improves arousal. This achievement could improve the quality of life for millions of women who suffer from sexual dysfunction. And while I had a lot of success this summer with in vivo tests, for long-term use in human patients, we may need to implant electrodes. This requires the development of electrodes that have minimal tissue interactions. And this is what my lab mate Chris has been working on this summer.
For my SHIRT project, I created nerve electrodes that are coated with an omniphobic material that limits tissue adhesion in order to record from and stimulate on the sciatic nerve. This material may improve the tissue response of the electrode, allowing for improved signal recording and stimulation thresholds. I mix the cuffs by gluing 3 stainless steel wires into a silicone tube with a longitudinal cut that allows for the tube to be placed around the sciatic nerve. Here's the final electrode before being coated. As you can see, the bubble of water has a larger external contact angle with the electrode. After coating, the electrode is more hydrophobic, so the water bubble has a smaller external contact angle in order to minimize contact with the liquid. Despite the coating, the impedance of the electrodes does not change. Here's an example of a stimulation trial where current was sent through the cuff in order to get a muscle response. The stimulation traveled fast and is shown on the left, and then evokes the muscle response on the right. We can also use the electrode to get neural recordings. During this trial, we flexed and extended the leg, as shown on a line in the body. This corresponds to a change in the level of neural activity, as shown above. Here's a picture of the electrode in vivo. We then took samples from the proximal to the cuff, in the cuff, and distal to the cuff, and analyzed the histology. Here's a cross-section of the sciatic nerve from the coated cuff. The larger dark regions are the fascicles which contain the nerve axons. This is the cross-section of the sciatic nerve from the uncoated cuff. The fascicles are still darker areas in the middle. In addition, however, the perimeter of the nerve has the darker tissue. We're in the process of determining whether this indicates that the uncoated cuff caused a larger tissue response than the coated ones. From those slides, I created a MATLAB code that calculates the area occupied by the axons in order to indicate any damage to the nerve. As shown in this graph, there is little difference between the coated and the uncoated cuffs in regard to the axon area.
Having the chance to participate in my own research project this summer has been an amazing experience. Next summer, I plan to start applying to PhD programs, and this has given me a great idea of what research will be like.
The SHIRT program has also given me a great research experience this summer, so I intend to continue my research through the school year and get a master's degree through the SUGS program here at Michigan. To end our video, I'd like to thank members of the P Neuro Lab for helping with our research and our video. I'd also like to thank the Instrumentation Lab for making the omniphobic coating that I used on my electrodes, and I'd like to thank the SHIRT program for giving us this great research experience. Thank you.
Thank you.